Hello, good evening to all. My voice is audible. Please let me know my voice is audible. Yes, good evening to all. My voice is audible. Okay, good evening, Shivangi. How are you, Bhavni? How are you all? I hope you all are fine and doing great in your life. How are you all? Please let me know. Yes, yes, thank you, Bhavni. So, how are you? So we have done some important topics in genetics. Even we have covered very important topics. And yes, I also gave you some homework also. So I hope all of you must completed it. But I'm not getting as much as assignments as it should be. Like we have 96 members in your batch, but the assignments are very few. So again, I request all of you to please submit your assignments on time so that you all will be free early and you will be focused more on your review paper. Okay, so I am just hoping that all of you will send me your assignments as soon as possible. Okay, so are you getting my voice? All of you, are you getting my voice? Okay, now just I am going to start the session because as we already discussed before also, we have to take little bit extra time so that we will able to do it on time. So from when you have your exam? From when? From 16th of December. Your exams are from when? Please let me know. From when will you have your exam? Please let me know. Yeah, Shivani. Please, Shivani. From 15th of December. Okay. I'm not getting Bhavni. What is basically NPTEL? Please let me know what is NPTEL. So some of you have from 22nd of December, but many of you have from 15th or 16th of December. No worries, we will try to complete our topics before this so that you will get ample time for your review paper also. And yes, even you will able to submit the assignments also on these times, on these days which are here before 15th of December. So I will try to wrap up your lecture series by 15th of December so that you will focus on your exam, and also you will able to work on the review paper also. So yes, we have to discuss today. I am just waiting 1 or 2 minutes more so that all others will join with us and we will start the session. So I'm just waiting for 1 or 2 minutes more, otherwise I will start the session. So yesterday we have done chromosomes and we have done mitosis and meiosis. I think mitosis and meiosis is your homework also, which you have to do. Now what happens now? Some students are submitting the older assignments instead of and even the you are paying the message which is not for the yesterday's homework, but it is before yesterday. So I request you to just pin that homework which we have to do. Like the homework which I gave you, you have to pin that particular homework. Okay. So yes, some of you have drawn the stages of mitosis and meiosis, but only few of you. Otherwise, some students are still submitting the assignment of semi-autonomous nature. Okay, okay. NPTEL provides e-learning through online web and video courses, various streams. Oh, good. It's good. Yeah. So they are just only few of you. Chandra is here. Chromosome, DNA, gene. Why mitochondria is semi-autonomous in nature? Types of chromosomes. And I just want more and more. You know, I just want that you send me more images of mitosis and meiosis, because mitosis and meiosis is quite important, but only few of you are able to submit it till now. We have Shivangi. Shivangi has done mitosis meiosis, very good. Yeah. Bhavni, you use colored sketches, okay. Orange and green, very good. But you only show me mitosis, but no meiosis. Bhavni, so I just request you all that thank you for your efforts, but I just want that you send me mitosis and meiosis again if possible. So this is your homework and you have to pin it. You have to draw the diagrams of mitosis and meiosis with the help of color sketch. Okay, even take one day more, no issues, no problem, but try to do it with perfection. So you have to pin this. Yeah. You have to do both. So this is your homework for today also. You have to pin this question, that diagrammatic representation of meiosis and meiosis. Mitosis and meiosis. So I will suggest all of you, if you want to take more time, instead of one day, keep 2 days. But mitosis and meiosis are very important, and I just want the very good diagrammatic representation of mitosis and meiosis. All of you, please. I request all of you that I want, this is your homework also for today also. You have to diagrammatically represent, or you have to make mitosis and meiosis diagrammatically with the help of colored sketches. So Bhavni, it's your duty to pin this message in the group so that all will be able to complete the assignment. Okay. So this is the homework which you have to do, because you are not doing it as much as I want, because I know you can do it. I expect little bit more from you. I'm sorry for it, but I know you all can do it. Definitely you can do it. Okay. So that's the whole thing. That is the thing which I want to share regarding the assignment. Okay, so Bhavni said these NPTEL is basically for the engineering students. Okay, so Bhavni, are you from engineering background? That means mitosis will be quite new to you if you are from engineering background. All of you, please, 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 all of you. Okay, okay, Shivangi. Okay, all of you, I request all of you to reply to me because I am unable to see any one of you. So your messages are the only one by which I will able to know whether you are getting me or not getting me. So please type, please, please, all of you. Okay, good happening, very good. So just keep it up, doing good work. You are all getting good things. I will try my best to so that all of you will get little bit more about genetics. Now we have to start, okay? Why you are not replying? You are 12 right now, but you are not, only one or two students are replying, and I am again asking all of you to reply because I am not able to see you. So your messages are the only one by which I will able to get whether you are getting me or not. Please, I request all of you to type some messages also. Shivani, you have to submit the check on my email ID. I already sent it before also. So you have to submit your abstract along with the topic on my mail ID. Okay, okay, good. Okay. So yes, now let me show you the topic which we have to discuss now. So this is our syllabus. So we have done Mendel's law of inheritance, we have done types of gene action, we have done quantitative traits, qualitative traits also. After it we have done ultrastructure of cell organelles, structure of chromosome, ultrastructure of cell organelles, structure of chromosome, morphology, karyotype, ideogram, and also we have done mitosis and meiosis. So these things are quite related, so I just covered these things before. So now we have these topics which are left. So this is regulation of gene expression, lack of brain, and fine structure of gene. So I will take these topics together because these are quite related. But before this I want to work on DNA structure function, DNA structure function, RNA structure function, DNA replication. So I will take these topics now, 12, 13 and 14. So I will try to cover these topics which are quite related. We have DNA, we have RNA. So I want to start these topics today. So we will start with DNA. How many of you are aware about DNA? What is basically DNA? Let me know. Any one of you? Any one of you? Please let me know. And yes, one more thing which is there. I just maybe I just didn't send you the PPTs till now. I'm sorry for it. I will send it right. Okay. Yeah. So you have to let me know about the DNA. What is basically DNA? DNA we have to do today, and we will try to cover RNA also. So DNA and RNA we will try to do today. Tomorrow we will try to do replication, transcription and translation. This is also known as central dogma. Yeah, deoxyribonucleic acid. Very good. So DNA is deoxyribonucleic acid. Just 1 minute. So I will share all these PPTs with all of you. I'm sorry for the delay. So we will study DNA today because some students are from another stream. So I will try to keep it very basic. Okay, I will try to keep DNA and RNA very basic so that all of you will be able to understand what is DNA. DNA is a genetic material. What is the functioning of DNA? What is the structure of a DNA basically? Okay. So now I'm just going to start the session. So we have to discuss DNA structure and its function. Now what is basically DNA? So it was in 1869. In 1869, there was a scientist whose name is Mr. He discovered nucleon. He discovered which is now known as DNA, but at that time known as nucleon in the cells from the pus cells, and later he separated it into a protein and an acid molecule. It came to known as nucleic acid after 1874. So in the 18th century, by some scientists, they are working on the nucleon basically. They will able to separate them as a protein and as an acid molecule. That is why the term is coined nucleic acid. Then in 1926 we have another scientist who proposed tetranucleotide theory, which states that nucleic acid consists of only 4 nitridases. It gives 4 different nucleotides on hydrolysis. So these are some proposed theories only. uh, uh, she is the lady Franklin, who used X-ray crystallography to visualize structure of a DNA. So she was the one who was able to visualize the structure of a DNA by using X-ray crystallography. Then we have very important or very famous scientists Watson and Crick. Watson and Crick, they were able to tell us or let us know about the structure of a DNA. So they proposed that DNA is double helical structure. They are the two scientists who was able to let us know that DNA is double helical structure. So when we are working on DNA, three scientists, number one is Franklin, along with Watson and Crick, their name will always came. So now what is basically DNA? Now the question arises, this is history only. Now what is DNA? So DNA is found in the cells of all living things. So this is the cell, like cell organelle. So in the cell we have nucleus. Inside the nucleus we have the DNA. So we have cytoplasm here, endoplasmic reticulum here, mitochondria here. Everything are there in the cell. So in the nucleus DNA is present. So DNA contains all of the genetic information that makes you who you are, and every individual organism has a unique DNA like a fingerprint. So you have to keep this thing in mind that every individual has a unique DNA. Every one of you. DNA is a genetic information and everyone has a unique DNA. So this is the cell, eukaryotic cell inside the nucleus. Inside the nucleus we have chromosomes, which we have discussed yesterday. And in the chromosomes, if you are able to study chromosomes, we have DNA along with the histone protein. So DNA is bounded along with the histone protein. In general, DNA is double helical structure. So DNA is double helical structure as proposed by Watson and Crick. So it contains phosphorus in the form of phosphate. DNA is known as deoxyribonucleic acid. So D means deoxy, N means nucleic, A means acid. So deoxyribonucleic acid. It is a molecule that codes the genetic instructions. Most DNA molecules are double-stranded helices. Each molecule consists of two long biopolymers made up of simpler units called nucleotides. Each nucleotide is composed of nucleobase recorded using the letters GATC. DNA is well suited for biological information storage. So DNA is basically the genetic material which contains the whole information. So again, DNA stands for deoxyribonucleic acid. This term was coined by Zacharias. So DNA is a biopolymer consists of nucleotides. DNA is a double helical structure in eukaryote and prokaryote. So DNA is a double helical. So like this is double helical structure in case of eukaryotes and prokaryotes. But in viruses, it may be double-stranded or single-stranded. So basically the DNA is always double helical, but sometimes it is exception in case of viruses, it is single-stranded. So in eukaryotes, DNA is presented in the nucleus as we have discussed. Sometimes it is also present in the cytoplasm. Like in case of prokaryotes, that means bacteria, the DNA is present in the cytoplasm. So DNA is basically what? Deoxyribonucleic acid, number 1. It is the hereditary material in the humans and almost all other organisms. So DNA is the hereditary material. DNA is the hereditary material in the humans and nearly every cell in a person's body has the same DNA. So in your body, if you have so many cells, so every cell has same DNA. Now where it is located? DNA is located where? Inside the cell we have a nucleus and inside the nucleus DNA is located. So most DNA is located inside the cell nucleus, but sometimes it is present in the mitochondria also. That is why mitochondria is semi-autonomous in nature because mitochondria has its own DNA. That means it has its own functioning. And sometimes it is present in the cytoplasm also in case of viruses or bacteria. Like in prokaryotes, the DNA is present in the cytoplasm. Now we have to understand the DNA structure. So the structure of a DNA is illustrated by a right-handed double helix with about 10 nucleotide pairs per helical turn. Each spiral strand composed of a sugar phosphate backbone and attached bases is connected to a complementary strand by hydrogen bonding. Let me show you. Yeah. So this is basically the structure of a DNA. DNA consists of deoxyribose sugar, which is a pentose sugar. That means a 5-carbon sugar. Along with this sugar, we have phosphate group attached on the one side, and on the other side we have the base attached. So this is the pentose sugar in which we have the phosphate group attached on the one side, and on the other side we have a base attached. So this is the pentose sugar. We have 1, 2, 3, 4, 5 carbon sugar. Now sugar is of two types. One is deoxyribose sugar and another is ribose sugar. Now what is the difference between ribose sugar and deoxyribose sugar? Ribose sugar is found in RNA, deoxyribose is found in DNA. So in case of deoxy, all the rest of the structure is same. The only difference is here. So in the ribose we have OH group attached, and in case of 2-deoxy, OH group is removed and only H is present. So we have two types of sugar. We have simple sugar, ribose sugar, as well as we have 2-deoxyribose. So here the OH is present, but right here we have OH group is not there, only H is present. Rest otherwise the structure is same. After it we have the another thing which is present in the DNA, which is the base. base pairs basically. Now what are bases? So they are divided into two groups. We have pyrimidines and we have purines. Pyrimidine is basically thiamine and cytosine. Purines are basically adenine and guanine. So I already discussed it in my biochemistry batch in very detail. So if you will not able to understand right here, you will have to go to that link also to understand DNA in detail. So here what happens, the bases are, we have two types of nitrogenous base pairs. We have two types of nitrogen bases. We have pyrimidines, which are single ring structure, only a 6-member single ring structure. In it we have thiamine and cytosine, and in case of purines we have two ring structure. One is 6-member and another is 5-member, and the example is, and the basis which we have here, adenine and guanine. So let me show you now. If you are able to understand here, this is the DNA. Right now we have to focus only on DNA. So this is the ribose sugar. If you are able to see, on the ribose sugar, on one side we have a base attached, and on another side we have a phosphate group, POOO. So we have a phosphate group attached. So this is the basically the DNA. We have a ribose sugar. On the one side we have bases, that means adenine, guanine, cytosine or thiamine. And on the other side we have a phosphate group attached. So similarly here you will not have to see this RNA right now. Only focus on this. So this is the ribose sugar. In the one side we have a base attached, and on another side we have the phosphate group attached here. Similarly, this is again the ribose sugar. We have deoxyribose sugar. We have base attached, and on another side we have the phosphate group attached. Then again sugar, base, and the phosphate group. So this is basically the chemical structure of the DNA. We will discuss about RNA later on. So there is one more term which is there in in the DNA and RNA, nucleotide and nucleoside. So this is your homework. You have to tell me the difference between nucleotide and nucleoside. I will also tell you in this PPT also, but again you have to tell me in detail. So these are the bases which we have to study. We have to study adenine and guanine, which are the purines, which is the double ring structure, and we have to study cytosine and thiamine, which is single ring structure, so pyrimidine. In case of RNA, instead of thiamine we have a uracil, which is the base basically. So these are the nitrogenous bases which are there along with the sugar molecule, along with the phosphate group. We have basically the pyrimidines, cytosine and thiamine, and the purines are adenine and guanine. These are the nitrogenous bases. So on the one side we have the base attached, that may be either cytosine, uracil or thiamine, or on the another side we have a phosphate group attached. So are you getting it up to now? Is it fine? Please let me check it. Are you getting it? Now the another term which is there is tautomerism. Now what is basically tautomers? They are the isomers of a compound which differ only in the position of protons and electrons. A reaction which involves simple proton transfer in an intermolecular fashion is called tautomerism. So we have keto-enol tautomerism is a very common process and is acid or base catalyzed. Basically what is keto-enol tautomerism? Aldehydes, ketones and some other compounds undergo this type of tautomerism. So from the keto form we are able to get the enol form. Okay, so there is a migration of protons from the alpha carbon to the carbonyl oxygen. So migration of proton is there, and from the keto form C double bond O we are able to get the enol form. So this is the keto form of thiamine, the base pair which we have, and this is the enol form of that particular thiamine. Now yes, this is basically nucleosides and nucleotides which you have to tell me. We have nucleosides and we have nucleotides. Now what is the difference between both of these? What is basically nucleosides? When we have the 5 carbon sugar and we have only base attached. That means we have either adenine, guanine, uracil, cytosine attached with the 5 carbon sugar. Then it is known as nucleosides. So when adenine is binded with the ribose sugar, it is known as adenosine. Guanine is bind with the ribose sugar, it is known as gynosine. Similarly, adenine is bind with the deoxyribose in case of DNA, it is known as deoxyadenosine. Guanine is bind with deoxyribose, it is known as deguanosine. So this is nucleosides basically. So you have to draw a diagram of nucleosides as well as nucleotides. Now what will happen in case of nucleotides? We have the sugar molecule, we have the ribose sugar or deoxyribose sugar, along with it we have a base pair attaches, and we have also the phosphate group which gets attached. So this is the nucleotide. Now what is adenosine now? So adenosine is basically what is adenosine? Adenine plus ribose. Now plus add phosphate. So when we are adding the phosphate along with it, what we will get? We will get adenosine monophosphate or AMP. Similarly, we have guanosine. So when with the guanine we have a ribose plus we will add phosphate. So what will it will become? It will become gynosine monophosphate. Similarly, uridine is known as uridine monophosphate. Okay, so you have to tell me the difference between nucleosides as well as nucleotides. Now what is the function of these nucleotides? Nucleotide is basically what? Ribose sugar, base is plus phosphate group. So nucleotides are the precursors of nucleic acid, deoxyribonucleic acid and ribonucleic acid. The nucleic acids are concerned with the storage and transfer of the genetic material. So so if you are able to see, there is a double helical structure inside the chromosome. So the nucleic acids are concerned with the storage and transfer of the genetic information. The universal currency of energy, namely ATP, is a nucleotide. So we need ATP for the activity of the cells. So nucleotides are composed of components of important coenzymes like NAD and FAD and metabolic regulators such as CAMP and CGMP. So DNA must be stable. They are the coding and non-coding functions which are found in the DNA. So this is basically the terms which are there in case of DNA structure. We have nucleosides as well as we have nucleotides. Now what is basically a DNA? DNA is a polymer of deoxyribonucleotides. So when these nucleotides they join, it becomes polynucleotide chain, that is known as a DNA. When nucleotides, like here, if I will show you here, so these nucleotides they are just joined with each other, they are forming a chain or a chain of a nucleotides, polynucleotide chain. This is known as DNA. This is known as DNA. So similarly here, DNA is a polymer of deoxyribonucleotide. It is found in the chromosomes, mitochondria as well as chloroplast. It carries the genetic information. So DNA is basically what? It is the polynucleotide or it is a polymer of deoxyribonucleotides. We consist of base, sugar and phosphate in case of nucleotides. This is a ribose sugar. On the one side we have either purine, pyrimidines, and on another side we have a phosphate group which is attached. So you have to draw the structure of nucleosides as well as nucleotides along with all purines and pyrimidines. So these are the pyrimidines which is single ring structure, and these are the purines which is adenine and guanine which is the double ring structure. So pyrimidines are cytosine, thiamine and uracil. Purines are adenine and guanine which is the double ring structure. Again this is the same thing. What is basically adenosine and what is adenosine form of? So it consists of basically only the sugar base along with the pentose sugar along with the base only, either adenine. So if it is adenosine, that means the base which attached is adenine. So this is the structure. This is a polynucleotide structure. we have a ribose sugar. On the one side we have a base pair, on the another side we have phosphate group. Similarly, ribose sugars, one side we have the base pair, on the other side we have again the phosphate group. So you have to draw the structure of this DNA also. So we have this sugar. On the one side we have the base pair like guanine, thymine, adenine, cytosine, and on the another side we have the phosphate. Okay, so the secondary structure of the DNA is double helical. As you all know that the DNA is double helical structure as proposed by Watson and Crick. So the secondary structure of a DNA, two antiparallel polynucleotide chains would around the same axis, bound around the same axis, sugar phosphate chains wrap up, and the the base pairs which are here, we have adenine, thymine, cytosine and guanine, they will form the hydrogen bond. Let me show you. Yeah. So if you are able to see here the nitrogen base pairs which we have like adenine, cytosine, thymine and guanine, they will form the bond. So adenine always pair with the thymine. Getting my point? So adenine always pair with the thymine with two hydrogen bonds. Cytosine always not with two hydrogen bonds. 1 minute please. So adenine always pair with thymine, cytosine pairs with the guanine. So we have this type of interaction or this type of base pairing within these nitrogenous base pairs basically. So in case of adenine and thymine, we have a double bond. In case of a cytosine and guanine, we have a triple bond. So you have to keep in this mind. Adenine forms the hydrogen bond with thymine. There is two double bonds. And in case of cytosine and guanine, the presence of three double bond, hydrogen bond. So by this, they are able to form the bond. So base taking the bases in the DNA are planar and having a tendency to stack. Major stacking forces are hydrophobic interaction van der Waals forces. So this is the thing I want to show. So that this is a double helical structure which is in the DNA. We have major group as well as we have the minor group here. Now this is the types of DNA. We have A DNA, B DNA, Z DNA. So Z DNA is the most common type of a DNA. This is according to the helix size, helix base pairs per turn and the diameter. According to this we have three types of DNA. So this is how the DNA molecule is surrounded or wrapped by the histone protein, and this wrapping of the proteins and the DNA is known as nucleosome. So nucleosome is any of the repeating globular subunits of a chromatin that consist of a complex DNA and histone. So there is a bonding of the DNA and the histone proteins. It is known as nucleosome. So if you are able to see here, so this gray one is and this blue one is the histone and the DNA protein. So this blue one is basically the histone protein. So this is how we are able to get this. The DNA is double helical structure and then it wraps around the histone protein and it forms inside the chromosome. So this is how it looks inside the chromosomes basically. Like this, the DNA is very much supercoiled in nature. Then this DNA gets denatured also. What is basically denaturation? When from the double stranded, from the double helical, we are able to get single stranded structure. This is known as denaturation. So either by heating or by just giving them the temperature, we are able to denature the DNA molecule. Now this is a function of DNA that it consists of four bases: adenine, guanine, cytosine and thymine, that are held in linear array by phosphodiester bonds through the 3' and 5' position of the adjacent deoxyribose moieties. So DNA is organized into two strands by the pairing of bases of adenine to thymine and guanine to cytosine. So within adenine and thymine we have a double bond. Within cytosine and adenine and thymine we have a double bond. Cytosine and guanine we have a triple bond. So this is again the same thing, you have to read it by your own self. Now this is the difference between pyrimidines and purines. Like purine is single ring structure, purine is double ring structure, which is adenine and guanine, and pyrimidine is a single ring structure. So these are scientists Watson and Crick who was able to tell us about the DNA, double helical structure of a DNA, and they also got the Nobel Prize. So this is the double helical structure of a DNA, and yes, the bonds are formed in between the nitrogenous bases which we have. We have adenine, cytosine, thymine and guanine. So the bonds are formed in between the bases which we have. These are the forms of DNA, A DNA, B DNA and Z form of DNA, and the difference is only according to the this wrapping, according to the number of turns, the difference is there. So according to the coiling, according to the base pairs, the difference is there. So this is basically the structure. If you are able to see, like guanine forms a bond with cytosine, adenine forms the bond with the thymine. So purines and pyrimidines, they are forming the bond with each other. If you are able to see, like we have double bond between the nitrogen base pairs, even we have the triple bond. So between guanine and cytosine we have the triple bond, and between adenine and the thymine we have the double bond. Again, between adenine and the thymine we have the double bond, between cytosine and the guanine we have the triple bond. So there is the two polynucleotide chains, they are just joined together by the help of these nitrogenous base pairs only. So adenine and thymine we have a double bond, cytosine and guanine we have a triple bond. This is according to the Chargaff's rule. So otherwise the ribose sugar is there, everything is there. With the help of these nitrogenous bases, we are able to form the double helical structure. We are able to join the two different polynucleotide chains. So with the help of these nitrogenous bases, we are able to join the two different polynucleotide chains. These are again the forms of DNA. I am not going in detail. This is again the denaturation, when from the double helical structure we are able to get the single helical structure. This is known as the denaturation basically. So up to now, is it clear to all of you? I hope all of you, some of you are quite new to DNA. I hope you get it. Is it clear to all of you? Otherwise, then after it, I have to start RNA also. So that both these are quite related with each other. That is why I want to do it. And one more thing, please let me know the syllabus, not the syllabus, the homework also. Please let me know the homework which you have to do. The very first thing which you have to do is to do the you have to draw meiosis and mitosis, number one. What is the second thing which I asked you? The difference between the nucleotide and nucleoside. You have to draw this thing, and then thirdly you have to draw the DNA structure also. Getting it? So please, any one of you, please write it somewhere, so that after the end of session you have to write it in the group also. Yes, yes, yes. Thank you. You have to draw a diagrammatic representation of mitosis, meiosis, nucleotide, nucleosides, and also you have to draw the structure of a double helical DNA. Yes, yes. Now I am just moving forward to the RNA part which we have. So we have RNA, its structure and function. So like we have DNA and its function and structure, similarly we have RNA. Replication we will discuss, we will do it tomorrow. So I will just now do RNA. So just give me a little bit more time, then this will be over. Okay. So this is the RNA and RNA structure and function. You already know what is the difference between DNA and RNA. The only difference is between the sugar. Like in case of DNA we have deoxyribose sugar, and in case of one more difference is in the pyrimidine. So instead of thymine, in the RNA we have a uracil. So these are the things we already discussed. Nucleic acid is basically DNA and RNA. Nucleotides and nucleosides we have just discussed now. Nitrogen bases which are basically purines and pyrimidines. Pentose sugar is oxyribose sugar. or simple ribose sugar as well as deoxyribose sugar. So this is again they are starting it from at the very basic. So nucleic acids are the polymers of nucleotides. They include DNA and RNA. So nucleic acid, which was in the very early of the history known as nuclein, it is basically DNA and RNA. It is found in all living systems. Nucleotide. Tide consists of base, sugar plus phosphate group. Nucleotide. Side consists of only Side consists of only pentose sugar and nitrogenous base, but phosphate group is absent there. So nucleotide, which basically consists of pentose sugar, which may be ribose or deoxyribose, nitrogenous bases, which are purines and pyrimidines, along with the phosphate group. So this is the nucleotide. Again, we have purines and pyrimidines. Now we have to discuss on RNA. Sorry. So RNA, the backbone is sugar and the phosphate group. Nitrogenous bases linked to sugar moiety project from the backbone. Nitrogenous bases are linked to pentose sugar through N-glycosidic linkage to form the nucleoside. Phosphate group is linked to 3' OH of the nucleoside through phosphoester linkage. Two nucleotides are linked through 3,5-phosphodiester linkage to form a dinucleotide. More and more such groups will be linked to form a polynucleotide chain. Such a polymer has a free phosphate moiety at the 5' end of the ribose sugar and it is called 5' end of polynucleotide chain. At the other end, ribose has a free 3' OH group which is called as the 3' end of the polynucleotide chain. So in RNA, every nucleotide has an additional OH present at the 2' position of the ribose. So let me show you the diagram. So this is basically RNA, which is known as Mummy, aap bana do. Okay. So ribonucleic acid is RNA is one of the major biological macromolecules that are essential for all known forms of life. It is a biological macromolecule. Each nucleotide in RNA consists of a ribose sugar, as we already discussed, along with adenine, cytosine, guanine, and instead of thymine we have a uracil here. Instead of thymine we have a uracil. So as DNA is single-stranded structure, so we will have this ribose sugar with the phosphate group, and we have adenine, guanine, cytosine, and uracil. So this is a single stranded structure. So the synthesis of RNA. So synthesis of RNA is usually catalyzed by the enzyme which is RNA polymerase by using a DNA. So RNA is basically synthesized from the DNA. DNA is double stranded, from which we are getting the single stranded RNA. So by using a DNA template we will synthesize the RNA. This process is known as transcription, which we will have to discuss in detail. There are also a number of RNA, dependent RNA polymerase, that uses RNA as their template for the synthesis of a new strand of RNA. A number of RNA viruses use this type of enzyme to replicate their genetic material. So these are the types of RNA. We have tRNA, RNA and mRNA. So we have transfer RNA, messenger RNA as well as the ribosomal RNA. So we have three types of RNA: transfer RNA, messenger RNA and ribosomal RNA. Now what is basically the messenger RNA, which is also known as mRNA? Messenger RNA carries the information about the protein sequence to the ribosome, the protein synthesis factories in the cell. So basically this mRNA, they carries the information from the DNA only. So messenger RNA from the nucleus, they will pass the information to the cytoplasm. So they carry the information to the ribosome, the protein synthesis factory in the cell. So from the nucleus, we will able to get information into the cytoplasm. It is coded so that every 3 nucleotides correspond to one amino acid. So in eukaryotic cells, one precursor mRNA has been transcribed from DNA It is processed to mature mRNA. This removes its introns, non-coding sections of the pre-mRNA. The mRNA is then exported from the nucleus to the cytoplasm where it bonds with the ribosome and they later will form the protein. So two terms are there, transcription and translation. From DNA we will get RNA and then from RNA we will get the proteins with the help of these mRNAs. Now another type is ribosomal RNA. So ribosomal RNA is the catalytic component of the ribosome. Eukaryotic ribosome contains 4 different types of rRNA molecules, which is basically 18S, 5.8S, 28S and 5S rRNA. So the ribosome, this ribosomal rRNA is basically of 4 types. 3 of these are synthesized in the nucleolus and one is synthesized elsewhere. So in the cytoplasm, ribosomal RNA and the protein combine to form the nucleoprotein, which is called ribosome. Please keep this thing in mind that this ribosome binds with messenger RNA and it is helpful in the protein synthesis. I will let you know everything related to it during transcription and translation. Several ribosomes may be attached to a single mRNA at any time. Nearly all the RNA found in a eukaryotic cell is RNA, which is ribosomal RNA. So there are some terms which is ribosomes, protein synthesis. We will discuss all those terms later. Now the third type of RNA is transfer RNA. This RNA is quite helpful during the protein synthesis. The transfer RNA is a small RNA chain of 80 nucleotides. It transfers a specific amino acid to a growing polypeptide chain at the ribosomal site of a protein synthesis during translation. It has sites for amino acid attachment and an anti-codon region for codon recognition that binds to a specific sequence on the messenger RNA chain through the hydrogen bonding. I am not going in detail right now because we will have to discuss it during transcription and translation. But you have to keep in mind the types of RNA. We have messenger RNA, ribosomal RNA and transfer RNA. So this is double-stranded DNA. From this we will able to get the single-stranded RNA. And this process is known as transcription. Then we have some another types of RNAs also. We have small nuclear RNA. These small nuclear RNA are involved in the process of splicing of primary transcript to form mature mRNA. Because sometimes what happens, there are some unwanted sequences, like this is unwanted sequence which is known as intron. So we have to remove that unwanted sequence so that only exons will bind further. So for that purposes we need small nuclear RNAs. So these small nuclear RNAs, they help in the splicing. Then we have microRNAs. MicroRNAs, short non-coding RNAs present in all living organisms. They have been shown to regulate the expression of at least half of all human genes. These single-stranded RNAs exert their regulatory action by binding messenger RNAs and preventing their translation into proteins. So they have small microRNAs. They will not allow the RNA to change into the protein. So they will not allow them to change into the protein. These are microRNAs. Then we have small interfering RNAs. These are the types of RNAs basically. Now small interfering RNA are 20 to 25 nucleotide long double-stranded RNA molecules that have a variety of roles in the cell. They are involved in the RNA interferase pathway where it interferes with the expression of a specific gene by hybridizing to its corresponding RNA sequence in the target mRNA. So this RNA is basically 20 to 25 nucleotide long double-stranded RNA. Otherwise, it is the RNA should be single-stranded, but it is double-stranded RNA which is involved in RNA interference pathway. So this is quite helpful in the formation of a single-stranded RNA. Then guide RNA is here, complementary RNA. These are the functions of RNA, messenger RNA, tRNA, rRNA. These are the functions of some of these RNAs, like messenger RNA. Messenger RNA carries genetic information of a DNA for protein synthesis from nucleus to ribosome. So basically, messenger RNA carries the information from the DNA because DNA is there in the nucleus. So this will carry the information from the nucleus into the cytoplasm. Transfer RNA acts as adapter molecule, carries amino acid and drops it to the particular location by recognizing codon on mRNA by virtue of having anticodons. tRNA is basically for the protein synthesis. rRNA is basically involved in the formation basically in the transcription along with translation. So it makes complex with the proteins and form ribosomal subunits which provide space for the synthesis of single ribosomal RNA of smaller subunit helps correct orientation of mRNA during attachment. Then snRNA, miRNA, different RNAs are here. So you have to just go with these and you have to know the function of all of these. This is again the structure of RNA. So this is again is in your homework. You have to draw the structure of DNA as well as RNA. When you draw the structure of DNA, it should be double stranded. When you are drawing the structure of RNA, it should be single stranded. Like this is RNA which is single stranded and DNA is the double stranded. And yes, in case of thymine, we have a uracil carbon. In case of RNA, this is the difference between RNA and DNA. Kindly go through by your own self. Types of RNA we already discussed. I will share this PPT right now so that you will able to read it properly. These are the part of transcription and translation which I will not cover today. We will discuss it tomorrow. These are small nuclear RNA which just feeds the influence. This is again same thing. So I hope you all get it. Is it clear to all of you? Please let me know. I hope it is clear to all of you. Please let me know. Are you getting it properly? So are you getting it? Everyone is please reply all of you. Are you getting it or not? What is the obvious advantage of having RNA as a genetic material for transcription? Karan, please let me know basically what is transcription. You have to tell me what is transcription first. Transcription is when we are able to get the RNA from the DNA. From the double stranded, we are able to get the RNA. Now, what is the advantage? What you are asking basically? Genetic material is there, which is the DNA. The main genetic material is the DNA, and from the DNA we are able to get the RNA or the mRNA, messenger RNA. So this whole process is known as transcription. So in the process of transcription, both are involved. Even DNA is involved basically because from the DNA we are able to get the RNA. So there is no the question is not valid that what is the advantage of RNA. So in case of general transcription which is there is only a DNA. From that DNA we are getting a particular mRNA. and from that particular mRNA, from the process of translation, we will get the protein. Okay, so this is the two steps which is known as central dogma, and we have to discuss this tomorrow in detail. So I hope when we will discussing that thing, you will able to clear your doubt also. So I request all of you to do your homework, which we have to do. You have to draw the structure. If you want to take more time, Shivangi, you can message me personally on the Telegram. I will definitely reply. So yes, you have to draw the structures of DNA, the structure of RNA, nucleotides, nucleosides, mitosis, meiosis. If you want to take time, take one day more, but at least try to do it with perfection. Okay, so it's my request to all of you to please try to do it by perfection. Have you any another question or regarding the review paper also? Are you able to submit it by tomorrow or not? Please let me know. Yes, Shivangi, you can message me. I will definitely reply. Karan, you will be there tomorrow also for transcription and translation. Yes, have you done? Have you any query regarding? Have all of you sent me your abstracts or not? So you have to send it by tomorrow only. Okay. So we will meet tomorrow. Till then, take care of yourself. Okay, no issues, Urja. Just let me know about your topic only, so that I will able to know that you have done something. If you are not able to do the abstract, it's okay. But just send me the topic only if possible. Don't worry, Shivangi, Shivani, no issues at all. You can take more time, but please try to do it because when you will do it, then it will only be done. Okay. So the abstract word limit is 200 to 300 words. Yeah, 150 to 300 words are more than enough. Okay. Okay, okay, Urja. Okay, good, good. Yeah, yeah. You submitted me after I got it. Yes, you submitted it also. I know. Yes, yes, Ade, you can definitely make the changes up to at the last also. No worries at all. You can make changes. Don't worry. No worries. You can send me later also. So don't worry regarding it. You can send it to me later also. But try to do it because we have to do it as soon as possible. Okay. Okay. So I think that's all for today's session. I hope we will meet tomorrow now. So tomorrow we will try to cover, yes, tomorrow we will try to cover DNA replication transcription and translation. Okay, so we will try to cover these topics tomorrow. With this, we have only few topics which will be left. We will try to complete it on this Sunday. Possibly, we will try to by Monday or Tuesday, but I don't know how long it will take. But tomorrow we will do transcription and translation. Okay, so be ready with it. So till then, take care of yourself. Yeah. So good night to all of you. Please take care of yourself. If you have any query, you can ask me. If you have any other query, you can ask me regarding your review paper also. Yes, I have to send you the PPTs. I'm really sorry. I always forget. Now just right after this class, I have my one more lecture series. So I will try to send you by 10 p.m. or otherwise you have to remind me on your this genetics group also that ma'am please send. Yeah, I will be free by 10:15 p.m. or maybe by 10:30 p.m. I don't know. I will send it after it. Actually, I get so much tired after the classes and everything. So I just switch off the laptop and just go to sleep. So that is why I forget. No worries, I will send it definitely. I will try to send it today. Otherwise, in the morning by 9 or 10 p.m. you will definitely get all the PPTs. Okay? Yeah. So take care of yourself and please try to do the assignment on time. If you want to take 1 day or 2 days, no issues, take your time. But don't do it by hot and fast only, okay? Try it slowly and slowly but with perfection. So that's all from my side. Please take care of yourself. Good night. Bye bye. Take care. Okay, Bhavni, you are here. Okay, good. So yes, you all are welcome to be there, so that all of you, if you want to, get some extra knowledge, you are most welcome. You can even message me also right there, like maybe in your lecture series or maybe in MBPT lecture series, I have my students from my October batch, and they are messaging me that ma'am, how are you? We miss you. So I just say so you have to be there and you have to be messaging me. That is why I ask all of you to please message me so that I will able to revise your name. I will able to remind all of you. Like we have Nidhi, Ajay, Asri, Bhavni, Shivangi. So by just pronouncing your name again and again, I will able to remind all of you. So you are most welcome every time in every lecture series of mine. You all are most welcome. Okay. Thank you all for all of you to be here for your patience and everything. Take care of yourself. Good night and try to do the assignments. Okay, so I'm just closing it right now because I have another lecture series. So please take care of yourself. We will meet tomorrow. Bye bye. Good night.